So this chapter is about data visualization, and we're gonna look at the data in different ways. Also try to see what effect the quality control did with the data, and whether there is any biological signal in the data. So again, we are going to use Skater. It provides a lot of plotting functions, and we will start with the PCA plot. So PCA is probably the easiest way to quickly look at your data. And so here we have some details of how it's done. You can have a look. So basically, we just project the data on a plane where we can see the highest variation in the data. And that's the way basically to find signals in your data. So I'm not going into details. You can have a look what PCA is. You're probably all familiar with it. And we will start looking at the data now. So as you remember, at the end of the previous chapter, we saved the Skater object, and we created a log transformed counts matrix, if you remember that. So here we want to look at the data before the quality control. So we take the whole object here, looking only at the endogenous genes. So we're excluding the controls, ERCCs and mitochondrial genes. Also, we take into account the 500 top genes selected by Skater, basically based on the expression values. And we also color and size and shape the data by the cell annotation table. So this is how it looks like. And the expression values here are counts. So these are raw counts that we started with in the previous chapters. So as you can see, on this PCA plot, we have the first principal component, the second principal component, and we can see that the most variation is actually on the first principal component. 76% of variance is described by this component. And we can also see the bad batch that we identified during the quality control. And all other batches are kind of in the same cloud. What we also can see from this plot is that the variance is actually corresponding to the size of the dots on this plot, right? So the smaller dots or triangles or squares are here, and then once you go in this direction, they become bigger. And the size actually corresponds to the total features in our cells, right? And total features, if you remember, it's the number of expressed genes. So basically, this plot tells us that the variance in this data set, the highest variance, come from the number of expressed genes in your cells. And it's associated with the first principal component. This is probably not the most exciting result for you, because this is basically it's a basically technical reason for this plot to be like that, because, and it's usually the case in single cell data that the first principal component usually is associated with the number of expressed genes or the library size. So we will see in the next chapters what happens to this plot when we normalize the data by the library size and basically remove this effect of the library size. But for the moment, we just look at the raw data. So this is what it tells us. Now, if we use the log transformed counts, so the slot that we saved at the end of the previous chapter, you can already see that the variance became smaller on the first principal component. So it was 76%, now it's 43%. So it's almost half of the variance was removed by simple log transformation of your counts. So the effect of the library site is not as clear as it was before. And I think the individuals, if you look at the individuals which are represented by different shapes, they also become separated from each other. So basically, it tells us that simple log transformation is already quite useful thing to do with the single cell data. But it's not the, it's the minimal thing, but not what we recommend to do. So here we have this note in bold, which tells you that log transformation is not enough to account for different technical factors between the cells, for example, sequencing depth. So here we haven't normalized the data yet, so it's just simple log transformation. So we do not recommend to use just log transformed counts for your downstream analysis. We will show you what to use instead. But here we use it just for the presentation purposes, all right? So now let's look at the, so this data set was before the QC. So here we have all the cells and all the genes. So what happens if we use the data set after the quality control? So it's called UMI QC. So we plot the same data after QC. And as you can see, again, the variance on the first principal component become even smaller. So now it's just 17%. So quite a lot, because we removed bad cells, we removed the outliers. That's why there is less variance in your data. Also, you can see that this individual, which is, yeah, the individual represented by the squares become more separated from the other two individuals, which is kind of tells you that there is some signal. So the individuals, as expected, they probably should be separated from each other. And so now we have an exercise here asking you to plot the same data after the quality control. Instead of using 500 most variable genes, let's use all the genes that we have in the data set and how this changed the PCA plot. So if you do it, you basically just need to run this code and remove, or just instead of ntop equals 500, you need to put ntop equals 14,000 and something. And that's how the plot looks like after you run it on all the genes. And to me, there is no big difference. So it's a similar structure that we had here. So what it means is basically that the 500 top highly variable genes are enough to describe the whole data set, right? If you add more genes, you don't change the structure of the data much.
Is there any way for selecting, for example, the cluster using in the PCA, not just like by the coordinates, or you know what I mean?
Sorry, selecting cluster?
Select the cells that are clustering together in a PCA.
So you mean
just by coordinates.
So you want to find clusters in your data?
Yeah, but imagine that I'm seeing only in the PCA. So I see that there is a cluster, I don't know. It's not very clear, let's say.
Yeah, so
for example, like there, if you want to cluster yourself.
Yeah, so clustering, we will, the question is how can we find clusters in this plot? So the clustering chapter we will cover tomorrow morning, right? So basically there are several approaches. So you can use k-means algorithm, for example. You provide PCA coordinates to k-means and it will find clusters for you. There are other approaches, but we will cover it tomorrow. So at the moment we can find the clusters by looking at the plot, right? We are not doing it in an unsupervised manner. And then if we take only 50, the most highly variable genes here, actually it's becomes even more structured, I would say. So you have this individual here, this individual here, this individual still separated, but then this batch. separates from the rest. So the conclusion here is that the number of genes that you look at is important. Taking different number of genes, you might get different pictures from your data set. And that's it. So it's kind of overview of the PCA plots and the ways of doing it using Skater. Do you have any questions here? Yeah, so once you've done the clustering procedure, which we'll cover tomorrow, you can, well, for example, an SC3 tool that we use, it saves the clustering results to the same phenodata table. So you can then visualize your PCA and ask to show the clusters identified by SC3. So some tools which are integrated with Skater allow you to do that. If your tool is not integrated, then you can take the clustering results, basically cell labels. For each cell, you would have a label with which cluster it belongs to. And then you can manually save this column to your phenodata information, and then visualize it in the PCA and asking for this specific column from your phenodata. Okay, so the next visualization procedure that you can use, and which is very popular for single cell data, is t-SNE map. So t-SNE is abbreviation of T-distributed stochastic neighbor embedding. It combines dimensionality reduction with random walks on the nearest neighbor network to map high-dimensional data. So basically, it projects your data in two-dimensional space, trying to preserve the distances between the cells. But it is different from PCA. And also, you need to know that t-SNE is a stochastic algorithm. So if you run it 2 times, you will get different results. So PCA will give you the same results every time you run it. t-SNE is stochastic. And we find it not very useful sometimes to interpret your data. So for example, if you look at single cell papers recently published, there are lots of t-SNE plots. But what happens is usually people find the clusters before plotting the t-SNE, right? So then they know what groups they have in the data, and then they run t-SNE in a way that it represents the groups, and then they color code the groups on t-SNE plot. So they don't use t-SNE for clustering. They use it only for representation of the data. Because sometimes it can give you like the results, that preferable results that you want to get. Like PCA will not show you the groups, whereas t-SNE will. And that's why it's more preferable. But the problem with it is that, as you can see now, we'll see now, it's not really good for finding the groups in your data. So Skater provides the plot t-SNE function. Again, let's run it on the data set before the quality control. And here we provide the rand seed, the seed for the random number generator, just to make sure that if you run it, you will get the same plot. If you don't provide the seed, you can get different plots every time you run it. And here we already use log transform counts. So I'm skipping the step where we look at the raw counts. And basically that's what you get from t-SNE, which is nice. So it separates different batches from each other. But it's very hard to say whether you have any structure in your data. So you can say that, yes, the batches are separated, but also like this batch is split in 2 groups, right? The orange batch as well, which is actually the bad batch. You can still see some cells are here. So it's in a way, a complicated algorithm. So it's not easy to interpret. So then we run the same t-SNE on the data set after the quality control. and that's what you see. So probably similar to what we saw on the first plot. Maybe a bit different. So any questions so far? Sorry? Yes. Yes. So that's we have a question here, exercise 2, which basically asks you to run it with different perplexity value. So perplexity is one of the parameters that t-SNE algorithm is using. So not only that you get different results when you run it multiple times, it also you need to adjust the perplexity parameter. For example, here we run it with perplexity 10, and the results are a bit better than we saw before. So, and like you don't know in advance what is the good perplexity value. And we also provide a link here. If you go to this web page, you will see it's a very nice web page. It shows you how you can basically get completely different results from the same data using t-SNE and using different perplexities and just playing with it. It's a nice overview of how you basically you should not use it to find clusters in your data, to find groups in it, because you can get completely different results from the same data set. So here we have perplexity equals 10, perplexity equals 200, and they're quite different from each other. But in general, it keeps the structure that it identified originally, right? It's just moving the dots a bit, grouping them differently, but the structure is probably always the same. So the next chapter is the same exercise, but now we're going to look at the reads instead of UMIs. So the same way we did before. You run exactly the same functions, but using the reads, the scatter object that was created for your reads. So I'll give you some time, maybe 10 minutes, and then we'll go through this chapter quickly. So did you find any differences between the UMIs and the reads? Yeah? Yeah, so the question is how to choose. Yeah, so the question is what number of top highly variable genes to choose when you do PCA or t-SNE. So basically, you can start with the total number of genes that you have in your data set. If you want to, the point of taking only the top highly variable genes is that you want to remove even more noise that you have in your data. So we removed quite a lot of noise by quality control, but here we just, the less genes you take, the less noise you will have in your data. So if you trust the top 50, if you look at the genes and you think that the most important genes, you can take them. If you don't know those genes, so probably it's better to take a longer list of genes. That would be my answer. But in general, it's better to take everything into account. But sometimes you don't see the structure when you take all 14,000 genes, because the amount of information is larger and the noise in the data is larger. Whereas if you take only the few top genes, you might get some signal which you didn't see. So basically, I think it depends on the effect in your data. So if your biological effect or batch effect is strong enough, then you will see it the same way whatever number of genes you take, right? If it's very small and sensitive to noise, then depending on how many genes you take, you might get different results. The same applies to t-SNE. t-SNE is actually t-SNE is good when your groups are very well separated. So if separation is good enough, then t-SNE works nicely. If it's a subtle effect, then you might misinterpret your data by using t-SNE. So if we look at the reads now, so this is the row counts, and we are plotting again top 500 genes. Actually, you can see that it's a big cloud. Very hard to say anything about it. The only thing is that PC1 contains 6% of variance, whereas PC2 is only 3%. So basically, all your variance is actually in this PC. So the reads data is actually much noisier than the UMI data. That's one of the conclusions. But then if you just do minimal log transformation of your data, you can already separate some individuals from each other, and the variance becomes much lower. So that's always good to do log transformation on your single cell data. So what is the, this is the data after the quality control, so it reads QC. It becomes even more clearer now, and the variance is only 6% on the first principal component. But again, we can see that this individual separates from everything else, the same that we saw with the UMIs, which is a good result, so they kind of reproduce each other. Now we plot the t-SNE of log scaled, log transformed counts matrix, and this is very hard to say anything about. It's just a big cloud of points, I would say. So if we change, okay. So what did we change here? Yes, so this is the full matrix before the quality control, and this is after the quality control. Well, you kind of separate the individuals from each other, so it shows something meaningful. And then we change perplexity to 10 and perplexity to 200. So yeah, it doesn't really work as nicely as it did with the UMIs. So with the reads, t-SNE kind of struggles to find the groups, because the reads data is actually is noisier than the UMI data. And that's it. So we showed two approaches to visualize your data, which are the most common approaches. So PCA is used everywhere. t-SNE is very popular in single cell, but be careful with t-SNE. There are probably other ways to do it, which, well, there are, what are they called? Diffusion maps. You can also use for that, or pseudo-time trajectories. Depending on your data, if you, instead of having separate groups, you might have a trajectory in your data. For example, if you have differentiating cells in different stages of differentiation, then there are tools which can align your data on some pseudo-time trajectory and show you instead of showing the groups, it will show you the trajectory instead. So, but we'll cover those tools tomorrow as well. But these are the basic two approaches that you start with when you start analyzing the single cell data. Any questions? Okay, so let's move on to the next chapter. I think Davis.